Today's session is on enzymes in food processing and preservation. So it's not new that the enzymes are used for food processing and preservation. Enzymes have been applied in food processing and preservation for millennia. Man has used naturally occurring microorganisms, bacteria, yeast and molds and enzymes they produce to make foods such as bread, cheese, beer and wine. Food processing by using biological agents is historically a well-established approach. Today, enzymes are used in a range of applications such as bakery, cheese making, starch processing and production of beverages, etc. The enzymes offer number of benefits such as improved texture, appearance, nutritive value, it generates desirable aroma and flavors and digestibility. Despite this, in recent years, significant advances are taking place in enzyme engineering and biocatalyst design which have abundant scope in food production, processing and preservation. The main objectives of this course, after going through this session, you will able to learn about the food enzymes, their availability and mechanism of action in food processing and preservation. And you are also aware of various enzyme groups and applications in food processing and preservation. Okay, let's see what the enzymes are. So, you already know that enzymes are protein in nature that acts as catalyst in all living organisms, microorganisms, plants, animals and humans too. So, you know what are catalysts? Catalysts are compounds that increase the rate of chemical reactions in biological systems. As part of essential life processes such as digestion, respiration, metabolism, tissue maintenance, these enzymes speed up chemical reactions in biological systems. In other words, they are highly specific biological catalysts. Enzymes operate within a narrow set of conditions such as temperature, pH and are subject to inhibition by various factors. The enzymes are ideal catalysts as they work in more or less mild conditions. Hence, it is important, hence they can apply in food processing operations in which the manufacturer wants to modify the ingredients selectively without destroying the essential nutrients. Now, you can see the typical enzyme. So, coming to the nomenclature of the enzyme. The International Union of Biochemistry set up a commission in 1956 to propose a scheme of enzyme classification and nomenclature, and the proposed scheme is officially adopted in 1961. The principal division between enzymes was based on the type of reactions that they catalyzed. During 19th century, many enzymes were given names ending with "in", often followed by the name of the substrate or source of the enzyme. I will give you some examples like pepsin, pepine, physin, etc. At the end of 19th century, it was suggested that the use of suffix "ase" preceded by the enzyme substrate. This has been widely accepted and today it is used as a general method of referring enzymes in industry. I will once again give few examples for easy remembrance like proteases, peptidases, pectinases, etc. Often, the trivial name of the enzyme derived from the truncated substrate name with "ase" added identifies the substrate or substrate range better for the food technologist than does its systematic name or its IUB number or name. I'll tell you for easy remembrance for you students. For example, lipase is a trivial name for lipid hydrolyzing enzymes, but the official name and number of lipase is triacyl glycerol acyl hydrolase. The latter is precisely descriptive of what such enzymes do, and the term or number is vital for communication between the biochemist and the product chemist. I'll give you few more examples for easy application for food technology side. Okay, lipases. Lipases acts on lipids and break down to their component fatty acids and glycerol as far as the food scientists and technologists are concerned. This is the important information to be used in food product and process terms. And the trivial names of all food enzymes are used in food processing and preservation. Proteinases, usually shortened to protease, which acts on proteins by breaking the amino bonds that join their component amino acids into the protein polypeptide chain. I'll give you one more example. Lactase breaks down lactose, maltase breaks down maltose, pectinase breaks down pectin, and cellulase breaks down cellulose, etc. We can use number of examples like this. Classification of enzymes. Enzymes are divided into 6 major classes with several subclasses are there. The type of reaction, they catalyze, and the substance, sometimes we call it as substrate, the act upon is the basis of enzyme classification. So, I will tell you about, I just enlist all the classes of the enzymes here. The first one is oxidoreductases, transferases, hydrolases, liases, isomerases, ligases. This is the general classification of the enzymes. Coming to another very important aspect that we want to discuss is about unit of measurement, enzyme unit. You know, the determination of the amount of enzyme in absolute terms like grams, kilograms present or used in a process is very difficult because its purity is often low and a proportion may be in an inactive or partially active state. The activity of the enzyme preparation and the activities of any contaminating enzymes are the more relevant parameters which are usually measured in terms of activity unit. Generally, we use the letter U. Enzyme unit defined as the amount which will catalyze the transformation of 1 micromole of the substrate per minute under standard conditions. So, now we know the definition. Now, let us see the sources and the range of enzymes in food processing and preservation. Coming to the sources, there are 3 principal sources of commercial enzymes currently used in food processing, namely animal derived, plant derived, and microbial fermentation. The traditional source, if we speak about the traditional source, I already told you that enzymes are using from past. The traditional sources of the enzymes used in food technology have been the tissues of either plants or animals. Although these are still widely used in food production and processing and preservation. Food enzyme production and use towards microbial alternatives including genetically modified derivatives of these organisms or come into work. The largest group of commercial enzymes currently used in food industry is produced by microbial fermentation. But several animal derived enzymes and a small group of enzymes derived from plants are still being applied. I will give you a few examples of the enzymes widely sourced from animals and plants used in food processing. Table 1 shows the enzymes widely sourced from animals and plants used in food processing. If you see the enzyme alpha amylase, it's available from cereal seeds, for example, wheat and barley, etc. How it acts in the food is, it hydrolyzes the starch to oligosaccharides, and in food industry, it's applied for bread making, brewing, etc. If you take pepin, pepin is an enzyme which is available from the latex of unripe papaya fruit. The action in food is food and beverage protein hydrolysis. It's applied for meat tenderization, chill haze prevention in beer. And one more enzyme is bromelain. Bromelain is available from pineapple juice and stem. So, it is used in meat tenderization and it acts on muscle and connective tissue protein hydrolysis. And coming to lipase and esterase, it's also called as esterase. So, this lipase is available from gullet of goat and lamb, calf abamus, pig pancreas. It acts on triglyceride hydrolysis and it is applied for flavor enhancement in cheese products. Let's see about the enzymes which are derived from microorganisms used in food processing. Table 2 shows enzymes derived from microorganisms used in food processing. The enzyme is alpha amylase. The source of the enzyme is Aspergillus species and Bacillus species. So, action, if you see the action in food, it acts on wheat starch hydrolysis. It's generally applied for dough softening, increased bread volume, aid production of sugars for yeast fermentation, etc. And one more enzyme is pectinase. It's also called as polygalacturonase. The source of the enzyme is Aspergillus species, Penicillium, etc. The action in food is it hydrolyzes the pectin and it's generally used for various food applications like clarification of fruit juices by depectinization. Coming to mechanism of enzyme action, again it's more important for the food scientists and food technologists to know the mechanism, how actually it acts in the food matrix. The prime function of the enzyme is catalysis. We know that, isn't it? The nature of catalysis taking place in the biological system is similar to that of non-biological catalysis. Enzymes have one active site in the structure that have great specificity for certain substances and catalyze the transformation into specific products. Enzymes are usually very big in compared to substrates which are relatively smaller. The active site is the area on the enzyme where the substrate or the substrate attach to. The active site of an enzyme represents as the small region at which the substrate binds and participate in the catalysis. So, you can see the active site of the enzyme in the figure. The enzyme substrate complex formation. The prime requisite for enzyme catalysis is the substrate. We know that, right? It must combine with the enzyme at the active site to form enzyme substrate complex, which ultimately result the product formation. There are a number of theories explaining this. A few theories have been put forth to explain the mechanism of enzyme substrate complex formation. Lock and key model. This theory was proposed by German biochemist Emil Fischer. According to this model, the structure or conformation of the enzyme is rigid. The substrate fits to the binding site just as the key fits into the proper lock. Thus, the active site of the enzyme is a rigid and pre-shaped template where only a specific substrate can bind. This model does not give any scope for the flexible nature of the enzymes. Induced fit theory or Koshland's model. Koshland proposed a more acceptable and realistic model for enzyme substrate complex formation. As per this model, the active site is not rigid and pre-shaped like other model. The interaction of the substrate with the enzyme induces a fit or a conformation change in the enzyme, resulting in the formation of strong substrate binding site, right? And you can also see the mechanism, the total mechanism of enzyme action in this figure. Now we will discuss the most important part of the session, is application of enzymes in food processing and preservation. Enzymes have played an important role in food processing and preservation, that we know, right? So, they are playing an important role since ancient times. If you take the examples, the example of an enzyme used were in the production of wine and whiskey. Today, enzymes used in various food applications include in the production of sweeteners, bakery and confectionery, alcoholic and non-alcoholic beverages, instant foods, infant foods, fish meal, cheese, dairy products, egg products, vegetable oil, purees, spice and flavor extracts, liquid coffee, flavor development, meat tenderizing, and plenty of applications are there. It has been observed a rapid growth in enzyme usage in food processing and preservation in recent years to increase yields of extract from raw materials, to improve handling of materials, to reduce processing cost and improve the shelf life and sensory characteristics of the foods. The use of enzymes provides many advantages, including higher quality products, energy efficiency, safer working environment. The main advantages of using enzymes instead of chemical modifications are that enzyme reactions are carried out under mild conditions of temperature and pH and are highly specific, thus reducing the number of side reactions and byproducts. The enzyme is mixed with the food, allowed to catalyze the required reaction and then either retained with the food or inactivated by the heat. Some of the food applications of main enzymes, there are number of enzymes. I will just give a few enzymes which are most extensively used in food processing and preservation. The first one is carbohydrates. This group of enzymes hydrolyzes polysaccharides or oligosaccharides. The commercially important types under this group are alpha amylase, glucoamylase, invertase, lactase, glucose isomerase, etc. Coming to alpha amylase, it hydrolyzes alpha 1,4 linkages to liquefy starch and produce maltose. They do not hydrolyze alpha 1,6 linkages and therefore leave low molecular weight dextrins and oligosaccharides which are unhydrolyzed. Beta amylase are exoenzymes that remove maltose units from non-reducing ends of amylase chain. In amylopectin, the cleavage stops a few units away from the alpha 1,6 branches. Maltose syrups from barley or soybean starches are the products which have wide applications in, for example, I will say brewing, confectionery and bakery products. Okay, now let us see about another important enzyme, glucoamylase or amyloglucosidase. Glucoamylose is a saccharifying enzyme which removes successive glucose units from the non-reducing ends of starch molecules by rapid cleavage of the alpha 1,4 linkages and slower cleavage of the alpha 1,6 linkages. Unlike alpha amylase, which already discussed, it therefore hydrolyzes linear unbranched chains to form glucose. The products are sweet tasting, fermentable hydrolysates with special functional properties. So, these are used in the production of alcohol, ascorbic acid and penicillin, functional ingredients in dried soups, infant foods and sauces. Glucoamylose is used to increase the alcohol content and to reduce the carbohydrate content of the beer by converting dextrins to fermentable sugars during fermentation. It is also used in the production of vinegar and yeast from starch-based products. Now, let us see about the other enzyme, invertase. Invertase hydrolyzes sucrose to glucose and fructose. It is used to remove sucrose from foods to prevent crystallization in molasses and in the production of invert sugar, confectionery, liquors and frozen desserts. Okay, let us see another one, lactase. Lactase hydrolyzes lactose to D-glucose and D-galactose. It is used in the prevention of lactose crystallization in ice creams and frozen milk concentrates, in starter cultures for cheese and in production of foods for lactose intolerance people. Glucose isomerase is used for the production of high fructose corn syrup. This is a sweetener from glucose. Different proportions of fructose and glucose are prepared by adjustment of the incubation time with the enzyme. These products are widely used in an alternate to sucrose in the bakery and confectionery industries. Okay, let us see about the pectin enzymes and another important enzymes. 3 types of pectic enzymes used in food processing and preservation. One is pectin esterase, second one is polygalacturonase and pectin lyase. So, the functions of these enzyme are, it accelerates the rate of filtration of the fruit juices in fruit juice industry, and it removes pectin from fruit base prior to gel standardization in jam manufacturing, and it prevents undesirable gel formation in fruit and vegetable extracts and purees. It standardizes the characteristics of pectin for varied uses as thickener. It recovers citrus oils. It stabilizes cloud in fruit juices. So, like that there are number of applications for this enzyme. And we will see about cellulases and hemicellulases now. Fungal cellulase preparations, which contain smaller quantities of hemicellulase and pectinase, it act on the alpha 1,6 linkages of glucose units in soluble forms of cellulose. These are used to improve the filtration of the vanilla extracts generally, and it tenderizes vegetables prior to cooking. It degrades nutshells prior to oil extraction to save energy. It extracts flavor compounds from vegetables, which was previously difficult and expensive. Hemicellulases reduce the viscosity of several plant gums by conversion of D-xylans to xylo-oligosaccharides, D-xylose and L-arabinose. These are used as to reduce the viscosity of the coffee concentrates, as well as in the extraction and clarification of citrus juices. In the hydrolysis of apple and grape pomace to fermentable sugars, to increase the yield of essential oils, spices and other plant extracts. Proteases. Proteases are classified according to their pH optima into acid, neutral and alkaline types. Acid proteases are produced mostly by fungi, and neutral and alkaline proteases are produced by both fungi and bacteria. Neutral bacterial proteases, which also have significant alpha amylose and alkaline protease activity, are used to hydrolyze plant and animal proteins and to improve the flavor of crackers and handling of pizza doughs. Acid fungal proteases, which have significant alpha amylase activity, are used to hydrolyze gluten to reduce mixing time, to make dough more pliable and improve the loaf volume and texture. It also used to tenderize the meat and to prepare liquid meat products. It reduces the viscosity and prevents gelation of the concentrated soluble fish products. It reduces the setting time for gelatin without affecting the gel strength. Bromelain is a mixture of proteases which hydrolyze plant and animal proteins to amino acids and peptides. It is used to tenderize the meat and improve the handling of pizza doughs and chill-proof beer, producing of waffles, pancakes and wafers. Pepin and physin are proteases that have broad substrate specificities and stability at higher temperatures. They are used to produce savory flavorings for soups, to chill-proof beer and as meat tenderizers. Rennet. Rennet partially coagulates milk protein to form casein curds in cheese production. Calf rennet is expensive and the use of microbial proteases is therefore increasing. Oxidases. So, glucose oxidase oxidizes the glucose in the presence of oxygen to form gluconic acid. It is used to desugar and hence to stabilize egg products and to increase the shelf life of bottled beer, soft drinks, other oxygen sensitive foods. It has advantages over chemical antioxidants because it does not lose its activity over time and it is not itself oxidized. Coming to lipases, lipases hydrolyze fats and fatty acid esters to form diglycerides and monoglycerides. Preparations which contain smaller quantity of alpha amylase and proteases are used to improve the whipping properties of egg albumin, and it also used to modify or solubilize fats in breakdown of emulsions. It is used to improve the flavor of dairy products and for the production of free fatty acids from butterfat. These fatty acids are used in small amounts to enhance natural flavors, in large amounts to give buttery flavor, and in large amounts to give cheesy flavor. These are used in cheese flavor dips, sauces and snack foods, and it also used in processed cheese, soups, baked products, chocolates and other confections too. Other enzymes, there are number of other enzymes which are used in food processing and preservation. I just give you a few more examples at a glance. So, one particular enzyme which is used in food processing is diacetyl reductase. Diacetyl reductase, it converts the flavor compound diacetyl to flavorless acetonin to improve the flavor in beer. Enzymes are also used to debittering of fruit products. For example, I will tell you, lemoninase which hydrolyze the bitter component lemonin in orange juice, or naringinase which breaks down naringin in grapefruit juice. Pentosanase converts pentosanes to deoxylose and alpha arabinose to reduce bread staling, to reduce dough viscosity, to lighten the crumb of the rye bread and to prevent it from separating from the crust during baking. Now, let us see the advantages. There are plenty advantages of the enzymes in food processing and preservation. Let's see a few. Enzymes play a diversified role in various aspects of everyday life, including aiding in digestion, the production of food, and several industrial applications. Enzymes are used since ancient times to carry out important chemical reactions for making food products such as cheese, beer, wine, bread, and yogurt, to which it also owes their flavor and texture. The use of enzymes frequently results in many benefits that cannot be obtained with traditional chemical treatment. These often include higher product quality, lower processing cost, less waste, and reduced energy consumption. Many of the traditional chemical treatments are generally non-specific, not always easily controlled, and many create harsh conditions. They often produce undesirable side effects and waste disposable problems. The degree to which a desired technical effect is achieved by an enzyme can be controlled through various means such as dose, temperature, and time. The amount of enzymes added to accomplish a reaction is relatively small as they are catalysts. We know that. For example, an enzyme preparation in most foods uses is equal to 0.1% or sometimes less of the product. Enzymes used in processing are generally destroyed during subsequent processing steps and not present in the final food product. So, like advantages, there are few limitations in enzyme usage. We just see the limitations, what the limitations are in food processing. Consumer acceptance. Consumer acceptance towards enzymatic processed food is low for few food products. Some people are having conflicts towards enzymatically processed foods. Some enzymes are retained within the food after processing may cause allergies in humans sometimes. Formation of large amounts of byproducts, most probably the unwanted ones, which are not accepted by the food manufacturers or processors. Enzymatic processing of food is cost-effective processing method when compared to other traditional processing methods. Consumers unaware of the use of enzymes and negative attitude towards enzyme technology. Now let's summarize the session once again. Enzymes are the protein biocatalyst synthesized by the living cell. They are classified into 6 major classes: oxidoreductase, transferase, lyase, isomerase, and ligases. Initially, enzymes were given names ending with "in", often followed by the name of the substrate or source of the enzyme, and later it was suggested the use of suffix "ase" preceded by the name of the enzyme substrate. Enzyme activities are usually measured in terms of the activity unit, U, the letter U generally we describe. Animal derived, plant derived, and microbial fermentation enzymes have been used in various applications of food processing and preservation. Enzymes operate within a narrow set of conditions such as temperature, pH, and are subjected to inhibition by various means. An enzyme is specific in its action, processing site where the substrate binds to form enzyme substrate complex before the product is formed. The mechanism of enzyme action is explained by lock and key model and induced fit model. The major enzyme groups such as carbohydrates, proteases, lipases, and oxidases, etc. are used in various food applications include in the production of sweeteners, bakery and confectionery products, alcoholic and non-alcoholic beverages, instant foods, infant foods, fish meal, cheese and dairy products, egg products, vegetable oil and puree, spice and flavor extracts, liquid coffee, flavor development, and meat tenderizing etc.